Hello everyone. Today we will be concluding the lectures on T cell book. And I have already discussed how T cells travel to bone marrow, and from bone marrow how they are their journey to thymus and how they get educated in thymus, and then how these T cells are selected, how they get activated, and then clonal selection, and then proliferation. So today, in the concluding part, I will be talking about more about proliferation, what kind of proliferation happens, that we will be discussing today. So coming to first slide, that is, I have already, in the first slide, I'm telling you that, let me talk about the first slide going, and I'm going to last. No, let me talk on the first slide first. So in case of part 1 and part 2, as I have told you, that their journey to bone marrow, then education in thymus, and then they come to secondary lymphoid organs where they are maintained in circulation. So these T lymphocytes undergo heterogeneous proliferation whenever they are introduced in lymphopenic host. So what is this heterogeneous proliferation? It is two types of proliferation, that is homeostatic proliferation and spontaneous proliferation. Goal of both the proliferations are same, that is to eliminate pathogen and also eliminate transformed cells, that is tumor or cancerous cells. They have to eliminate it and preserve harmless common cell and normal cells. That is the goal of both type of proliferation. What happens in case of homeostatic proliferation? Homeostatic is the physiological process, is a more of a type of physiological process in which T cells are maintained. Their number and composition is maintained in the periphery of the T cell pool, that is maintained in the periphery of lymphoid organs. And this pool is composed of large and heterogeneous repertoire of naive and memory T cells. And they are this pool, the proliferation here is a slow process, and it is relatively maintained at a constant level. And these memory T cells and naive T cells, because these naive T cells have to receive a signal, and I have told all the process, how they get activated and how they are going to fight the pathogen. So in absence of pathogen, a homeostatic proliferation is maintained, that is a constant number is maintained between naive T cells and memory T cells, which have already encountered the pathogen and they are in the form of memory T cells. That kind of proliferation, a kind of balance between naive T cells and memory T cells, is maintained by homeostatic kind of proliferation, and it is very much interleukin 7 dependent. This is for students in going for entrance exams, that homeostatic proliferation is interleukin 7 dependent. While coming to spontaneous, that is another type of proliferation. This happens during pathologic conditions, that is there is an introduction of pathogen. And I have already told you that what signals are important, how T cell is selected, naive T cell is selected, and how it goes for proliferation, all this mechanism. And there is a role of MHC also, that is major histocompatibility complex. All this mechanism I have explained in previous 2 lectures, that is on T cell 2 and in T cell 1 and T cell 2, part 1 and part 2. So and MSc, I have put a whole lecture on MSc, so that is also very important. So you have to relate all those things and then understand this spontaneous proliferation. If you are coming directly to the slide, it will be difficult to understand. So this is a more rapid process, and when a T cell decided to take us, when it goes into a spontaneous proliferation, it can take 3 parts, 3 major roles it can take. That is, it can become effector T cell and target tissues and go and fight there. It can become a memory T cell and recirculate, which is maintained by homeostatic proliferation, and it can become a memory cell and reside in tissues. So this was all about T cells, and I have put it into 3 different videos. I hope you are going to learn in it, and I'm waiting for your comments and reviews so that I can improve myself. Thank you so much.